I would like to make a brief introduction to the paper Activation of Endogenous Human Stem Cell-Associated Retroviruses and Therapy-Resistant Phenotypes of Malignant Tumors. The term endogenous human stem cell-associated retroviruses, in short SCARs, defines several structurally and functionally diverse families of genomic regulatory elements derived from human endogenous retroviruses. SCARs activation is required for pluripotency induction and maintenance of human embryonic stem cells. Activation of SCARs plays an essential role in the maintenance of the naive pluripotency state, development of preimplantation human embryos, and nuclear reprogramming of human cells from differentiated phenotypes to the induced pluripotency state. Genome-wide microarray expression profiling of human malignancies conclusively established that tumors manifesting close resemblance to pluripotent and self-renewal genomic regulatory networks of normal stem cells, collectively defined as stem cell-like cancers, are more likely to fail therapy, progress toward metastatic disease, and cause death of cancer patients diagnosed with these tumors. Observations that human malignant tumors diagnosed in multiple organs consistently manifest molecular signals of SCARs activation has led to the hypothesis that SCARs activation in cancer cells indicates that these cancer cells may acquire stem cell-like properties. One of the main topics of this paper is the critical analysis of the experimental and clinical evidence supporting the validity of SCARs hypothesis of human cancer initiation, development, and progression. SCARs expression is suppressed by highly sophisticated epigenetic silencing mechanisms in differentiated human cells. It is proposed that when this epigenetic silencing code is deficient or broken, the pathway to development of stem cell-like cancer is initiated. Analysis of mechanisms of actions of SCARs-derived genomic regulatory sequences helps to understand how activation of SCARs-associated pathways and genomic regulatory networks may affect phenotypes of cancer cells and facilitate malignant progression. These analyses specifically highlight the potentially critical impact on malignant progressions of SCARs-derived human-specific regulatory sequences. Overview of evidence that SCARs activation may contribute to the repair of the double-stranded breaks of damaged DNA molecules explains why SCARs activation may contribute to the increased survival and therapy resistance of cancer cells. It also helps to understand the molecular mechanism of biogenesis of human-specific regulatory sequences. Finally, and perhaps most importantly, the exciting new diagnostics and therapeutic approaches are highlighted that will help us to detect SCARs activation in tumors and offer efficient, targeted therapies to cancer patients. I hope you will find this work both interesting and encouraging.